Hello students, how are you all? I hope you all are doing well. I am Mr. Prithviraj. As you all know, I am your 9th class biology teacher. In our last class, we have gone through egg experiment where we have done little kind of experiment where we have seen the egg is either swelling or not. That is our activity we have conducted. Today we will discuss how to prepare a plasma membrane. And after knowing how to prepare a plasma membrane, we will conduct a certain kind of experiment with that plasma membrane. And we will see what observations we get from that particular plasma membrane. And then we will go for our further classes in which we will discuss what is the importance of osmosis in our everyday life. So let's start our class. The main experiment today is what? Prepare a semi-permeable membrane by using egg membrane. Okay. Our aim of the experiment is, what is our aim of the experiment? To prepare we will prepare a semi-permeable membrane by using egg membrane. So what are the materials we need? As you all know, we must write what all materials we need. We need 3 beakers, okay. After that we need egg membranes, 2 egg membranes, okay. We need 3 beakers, we need 2 egg membranes. Then what we need? We need sugar, we need water, okay, and along with that we need a measuring jar, disposable syringe, cloth and tablespoon. We need a measuring jar, syringe, okay, we need syringe, and we need cloth and tablespoon. These things, you know, we need cloth, then these are the materials which we need to conduct this particular experiment. Now let us conduct the experiment. So students, in this experiment what we will do, we will take an egg, we will write this, take an egg, now soak it in dilute HCl solution. We will take both eggs, here we will take dilute HCl, okay, we will soak it for 4 to 5 hours, and after dipping 4 to 5 hours what we will see? We will see that a membrane is coming out, means egg will come, but the cell is not there. The cell is dissolved completely. Now we will wash it. We will wash it with tap water. Now we will clean it with the cloth. Okay. After cleaning, we will get only a rubber type of egg, means without cell egg. Okay. Now we will the work of syringe. Now I will put syringe in it. Now I will put syringe in it, and we will suck all the materials out from the egg. We will suck all the materials out, means we need to do a little puncture on that. So when all the materials will come out, now we got what? We got one plasma membrane, means one egg membrane. Again we will do with that also the same thing. Now we got 2 membranes. Now we got 2 membranes. This is what they look like. They are membranes. They both are membranes. Now what we will do? We will take series again. We will fill one membrane with water. We will fill other membrane with sugar solution. Okay? What we will do? We will fill one membrane with water, other membrane with sugar solution. Now after doing that, what we will get? Okay, we got like this. Now this is filled with water, this is filled with sugar solution. Okay. Now we will tie it with little bit of thread. Okay. Now what we will do? We will take 2 beakers. Okay. One beaker we will fill it with water. Other beaker also will fill it with water. Now this bottle is with water. Sorry, we will do that. One bottle we will fill it with water, other bottle we will fill it with sugar solution. Both is 100 ml. This is 100 ml. This is also 100 ml. See, one only with water 100 ml, other one only with sugar solution 100 ml. Now what we will do? The ball which contains water, we will place it in sugar solution. This is the egg membrane. Along with water, we will put in the what? Sugar solution. And the egg ball which contains sugar solution, we will place it in water. Just we did replace it, the position. Okay. Now we need to observe what exactly happening. Now we need to observe. We will leave this particular thing for several times, suppose, or we will keep it for 2-3 hours. Okay. After 2-3 hours, we need to come and we need to measure. However, at first water was this much here. Okay. Now water level has been decreased. Whereas the size of the ball increased. Now look at here. This water level was this much. Now the water level has increased. But the size of the ball decreased. So what we got from this particular thing? What we have learned from this particular observation? We learned that sugar molecules are too big to enter into the plasma membrane. Right? If this is plasma membrane, sugar molecules cannot enter, or sugar molecules cannot go out. Why? Because plasma membrane won't allow those things. So what will happen? Water need to go towards sugar solution. So here what happened? The filled water now, they went out into the sugar solution. And here what happened? The water which was outside, it came into sugar solution to make it more diluted, because it was concentrated. No? So to make it dilution, they came here. Is that clear? So here this is due to osmosis. Across plasma membrane, across semi-permeable membrane. Osmosis means what? The flowing of water content from lower concentration region to higher concentration region in presence of semi-permeable membrane. The same thing happened here. This is a semi-permeable membrane where they have conducted all these particular things. It allows only certain materials to pass through it. Yeah, we know that it is selectively permeable membrane, so it will allow only certain materials to pass through it. Okay. Next, what is the importance of osmosis in living organisms? What is the importance of osmosis in everyday life? Here a lot of points are there. We will finish it in this class and upcoming class. We will try to complete in both the classes. Let us see. First thing, osmosis, the process of water moving through a semi-permeable membrane that does not allow dissolved solid to pass. So what exactly is osmosis? Osmosis means what? The movement of water through semi-permeable membrane through semi-permeable membrane which won't allow dissolved materials to pass through it. Here what will happen? If you will consider a plasma membrane, what it is doing? It is allowing only water or small contents which are dissolved in water, but it will eliminate most of the things which will come inside, like solids, protein, sugar and all. It will keep outside only. Okay, it won't allow them easily. So this is what our first point is. Second point, osmosis carries nutrient-rich liquid across the cell membrane. It carries nutrient-rich materials across cell membrane. Here when we talk about materials, that is exactly liquids which I am talking about. It will only carry nutrient-rich liquids across the cell membrane. Okay. Next, it maintains the ideal concentration of solute within the cell. What? It maintains ideal concentration of solute across and within. Across and within cell membrane. Suppose inside our cell sugar solution is there, some kind of Golgi apparatus or some kind of proteins are there. It will maintain the solute particles inside ideally as it goes outside. It won't allow them to go out, or it won't accept excess amount. The amount which is comfortable for cell, that much amount only will be, that much only allowed into the cell or outside the cell. That's what the maintaining a perfect ideal condition of solid materials or solute particles inside and outside the cell. This is also an important work. It is done by our particular plasma membrane. Now there are several other explanations are there which we will see in our next class. Okay, in our next class we will see them also. And today you need to, I hope you have wrote all this stuff which you have seen while I am speaking also. You need to note them down. In our next class we will meet with other important phenomena, other importance of plasma membrane inside our body. And along with that, we will conclude our chapter with activity number 3. Okay, thank you.